when I talked to you, it meant a lot to me to be like, yeah, you're a guy who knows how to have a job in a real world context. And that's totally been useful when you've been in our lab. You pitch in on everything. You've been really supportive of the other members of the team, and you've been effective at solving problems like developing new protocols and everything. So I really appreciate that you kind of brought a lot of those skills into your work here. And today, Kyle's going to talk to us about drainage in Northwest Minnesota, where he's been doing his research up at the Northwest Research and Outreach Center. So I'll leave it to you for that, Kyle. Thanks for being here.
Okay, with that, let me get my swig of coffee from my Dunkin' thermos. So, do we want to record this? Oh, oh, it is. Okay. No. Thank you, Anna, for that kind introduction. Let me. You will. Oh yeah, that's fine. Lights, door, whatever. Make yourself at home. Okay. Bailey, are we full screen online? We're good. Okay, let me. Okay, let me kind of start my timer up here. Okay, again, Anna, thank you for that introduction, and thank you all for being here in person or online. Again, I'm Kyle Sherbine, and this is my master's defense looking at soil organic carbon responses following 2 years of subsurface tile drainage in Northwest Minnesota. So the outline here is a pretty traditional introduction, experimental design results, and then what all of this means and what we can take away from it in conclusions. Okay, we're gonna get rid of this. Hide video panel. Okay. Like I said, not technologically savvy. Okay, so jumping right into the introduction, maybe. Okay, so why are we looking at drainage? So clay soils are naturally poorly drained, and this is because of that slow infiltration and high water holding capacity. And so drainage is growing as a management technique to mitigate the effects of changing and irregular precipitation patterns. And so in the 2017 US Census of Agriculture, they estimated that approximately 60 million acres are drained. And to put that into perspective, the entire state of Minnesota is in that range of 50 to 55 million acres. And surely over the past 5 years, that number has only grown. And so drainage provides benefits that we can see here, or what could be benefited by is removing that excess water. But with this, we also have drawbacks. We have the potential for nitrogen and phosphorus losses impacting downstream water quality. But what I'm going to talk about today is that potential for increased mineralization of soil organic matter. And this is really getting at and after how we sequester carbon in soils. So when we install drainage, we are changing the soil habitat. We're changing the water table, we're changing aeration in soils, and ultimately, those two things really dictate where living organisms can persist. So if we take these microbes living up here in our surface horizons, we install drainage, we change water in soils, change the aeration. We're allowing them to live in places that previously they wouldn't have been able to get to for whatever limitations. And so why do we care about soil organic matter? Well, it's the backbone of a healthy soil habitat. It's a reservoir for nutrients. It creates physical structure, enhances infiltration. And so as we change the soil system, soil biota are likely to shift and adapt, and aerobic organisms are likely to begin to dominate. And so this is where we see the potential for that organic matter that was previously waterlogged or unable to be reached could undergo rapid mineralization and subsequent depletion in soils. And it's important to recognize that not all soil organic matter is the same. So it's a continuum of decomposing plant and animal materials. It also includes microbial cells and tissues. It's a heterogeneous mixture of different fractions that form and persist differently throughout soils. And so looking at these different pools will help us get a better understanding of that biological response when we look at how carbon pools are changing with drainage. And so it's to kind of take a step back here, it's important to note that we are only looking at organic carbon in this study. We are not looking at inorganic. So organic carbon and carbon might be used interchangeably. So looking at soil organic carbon, this is what we're talking about. And so traditionally it's measured in carbon stocks. And so what we're really getting after here, big picture, is are we mineralizing organic carbon in drained plots differently compared to undrained plots? And so what are we actually measuring in the carbon cycle to understand this? We're looking at potentially mineralizable carbon, which is an incubation, and that's getting after that microbial capacity to decompose organic carbon and turn it into carbon dioxide. And we're looking at water extractable organic carbon, which is an organic carbon microbial food source that's easily mineralizable and accessible. And so these two things are respectively proxies for soil respiration and active carbon. And so these two indicators, they change over short periods of time, and they have been shown to indicate how bulk carbon is changing over time. With this, sticking with this microbial things impacted by microbes, we're looking at greenhouse gases. So here we're looking at carbon dioxide as a microbial byproduct. So if we see changes in microbial community structure and we see changes in the environment, we see changes in how microbes are acting in the soil, we expect to see changes in their byproducts. We're also looking at particulate and mineral associated organic matter based on a size fractionation. And this is getting at short and long-term carbon storage in soils. And so when we look at POM and Mayon, we're classifying everything in our sample as one or the other. And then finally, we're looking at organic carbon stocks, which is done through combustion, understanding that amount of organic carbon in the soil, and then scaling it up with bulk density for a known depth. So that's kind of a lot and not a ton of background. So the way that this is kind of structured is that in the results, we're going to look at more background of what all these are, what they tell us, the methods, and then the results, just to kind of keep things close together and to keep, I know there's a lot of, it seems like moving parts throughout this study. So why do we need this research? And so if we take, if we look at this broad collection of literature, we know there are other studies that have shown management decisions impact carbon cycling. And so a lot of these studies are looking at cultivation, crop rotation, cover crops, and tillage. When we zoom in on drainage, how drainage is affecting soils, we see that we're looking at these soils that aren't our traditional arable soils that we see just an hour away with corn and soybeans. We see organic soils, we see rice paddies and wetlands. And then when we even focus in more on drainage and arable soils, we see studies looking at total carbon as opposed to these individual pools that are going to give us a more in-depth understanding of the response and looking at how these are changing from those baseline conditions. Yeah, and understanding if these changes occurring in the soil are abrupt and quick changes, or if there's slow, gradual changes we see over time. And so what we're really, the questions we're really looking at here are, how are different organic matter fractions responding to newly installed tile drainage? Is organic carbon mineralizing at a faster rate in drained plots compared to undrained? And is carbon lost as greenhouse gases different in drained and undrained plots? Okay, so experimental design. Again, this is kind of the method section, and we will look at methods for all of these carbon metrics later in the results. So we are up here in Crookston, Minnesota, indicated by that orange star on the top image at the Northwest Research and Outreach Center. And so on our field in that bottom image, we have two different soil series. Both are silty clay loams, and with these soils, we see organic matter at around 4%. That field in the bottom image is 60 acres total. So each of those 4 plots are 15 acres. We have 2 plots, 1 and 3, that are drained there in the blue, and then we have 2 plots that are undrained in the gold, 2 and 4. And so this is a randomized complete block design with 2 blocks. And historically, this field has been farmed as 1 unit. And in the past decade, there have only been soybeans and wheat grown on this field, with wheat in 2019 and 2021, and soybeans in 2020 and 2022 this year. Yeah. And then those tile lines are, they were installed in 2019, and they are 1 meter deep and 15 meters apart. For those of you who are like me and probably trying to do that conversion in your head, that's 3 feet deep, 50 feet apart. And so it's important to note that a number of different parameters can go into that spacing or depth, but ultimately these parameters are what a contractor in Crookston would recommend for local growers. And so we wanted this to be indicative of what growers would see with their field as far as spacing. So soil sampling, you can see we collect cores with a hydraulic Giddings probe. And soil sampling occurs 3 times a year. The next slide is a timeline kind of going over all of that. But we collect cores to 90 centimeters depth. And that is really where we are installing those tile lines. And so that 90 centimeter core is broken up into 3 depths, 0 to 30, 30 to 60, and 60 to 90 centimeters. And it's important to note that 1 soil core was used for all the carbon analyses. So we're looking at soil in the same core. A second core was collected either for bulk density, for when we need that undisturbed core to get an accurate measurement, or for the sake of having a backup core in case we run out of soil and we obviously can't go back in time and recollect it. So kind of going back to our layout, in each of these 4 plots, we have 16 points. We collect a subset of 4 from these 4 plots. So that would be the 4 stars. And so when we analyze these by drainage treatment, we have 8 points in drained sections, 8 points in undrained sections. So the sampling timeline of all this. So drainage is installed in fall of 2019. Within days of installation, we collected 2019 samples. And so those discrepancies we see in 2019, if we see significant differences, we treat those as natural variation that we would have in a field of this size and not treatment differences because drainage has not been utilized. So fall sampling occurs in October, right after harvest. Spring sampling occurs in April or sometimes later, depending on when the snow cover melts. And then summer sampling really kind of falls around that 4th of July holiday and kind of just depends how people are in town and available. So PMC and WEOC, we are sampling over all 7, we're looking at over all 7 sampling events. We know that these pools are variable with time. And so we're looking at these more often. POM and Mayon, our short and long-term carbon storage pools, we are sampling once a year in the fall. We're looking at carbon dioxide emissions once a week across the growing season, and that is occurring, that's starting right around the spring sampling time and then goes until after that fall sampling event. And then organic carbon stocks we're looking again at in the fall of every year. And so statistics were performed in R, and we evaluated the effect of drainage treatment, drained versus undrained, and time. And so we are looking at, or I should say, drainage treatment and time were fixed effects and then block as a random effect. And then we also used repeated measures to kind of help us with that field variability and look at how those points are changing over time. Because all of these metrics are highly influenced by depth, analyses were separated into each of those 3 depths that I mentioned. And then a significant level was reported at the 90% confidence level at P equals 0.1. So results. And so this is a long section. So hopefully to break it up, there are some breathers here and there. So it's not just graph after graph. So stick with me here. So beginning, precipitation. We for those of us who have been here the past 2 years, we all know. Are we ever going to stop talking about the 2021 drought? Probably not. So why is this important and how does it translate into soil moisture? It's important because soil moisture is one of those things that's impacting those conditions on a small scale for microbes. And so here we're looking at soil moisture in that top 15 centimeters. And so you can see here in 2020, that wet year, we have similar patterns with at the end of the growing season. We're starting to see this gap with undrained plots having a slightly higher moisture. So we see that drainage is having an impact. 2021, our drought year, we don't see a ton of variation in between drained and undrained plots. We see it mostly at the beginning and end, maybe a little bit in the middle, but similar to 2020, we see a pretty or similar trends over the growing season. Yield. Yield was affected by drainage. So in 2019, again, before those tile lines were installed, we see yield in drained and undrained plots that are pretty similar. When we look at 2020, when we had that wet year, we see we had significantly higher yields in the drained plots. When we look at 2021 and we look at that drought year, we have significantly higher yields in the undrained plots. And so why is yield important other than this is what a lot of growers care about and what they want to see impacted? Increased yield is correlated with increased residues. And when those residues are returned to the soil, those are our carbon inputs. Okay, so our first little carbon metric here, PMC. So This is also known as the CO2 burst test, if you are more familiar with that. But PMC, it considers the available carbon substrates in the soil in the microbial capacity to decompose that carbon into what is measured as carbon dioxide. And so higher values correspond to more biological activity and indicate healthier soils. And so because this pool is, or this metric is influenced by microbes, as we increase aeration and create likely better conditions in drain plots, we expect PMC in drain plots to decrease. What this looks like in the lab, soils are re-wetted to 50% water-filled pore space, and that is based off of bulk density. They are incubated for 24 hours at 25 degrees Celsius, those ideal conditions, and then measured there in the background. You can kind of see it tied up to the infrared gas analyzer. So what do we see with PMC? We see this overarching trend that PMC decreased with depth and time. So on the y-axis, we have micrograms CO2C. Along the x-axis, we have those 7 sampling events from fall 2019 until fall 2021. And so, and again, this is broken up into those 3 depths, going from left to right, 0 to 30, 30 to 60, and 60 to 90. Oh, and the red triangles are means for each box. So we see it from 0 to 60, we see a significant treatment year interaction, those top 2 depths. When we move into the 60 to 90 depth, we see only treatment was significant with slightly higher PMC in undrained plots. Again, that general trend that we're decreasing over time and with depth, but at those shallower depths at 0 to 30, we see overall larger PMC values and more variability, or a larger range, I should say. And so when we look at PMC and we compare drained and undrained plots within a given depth at a given year, we only see one difference, and this is fall 2019 at this, this top depth. And so again, because this is fall 2019, be it or shortly after drainage was installed, we assume this to be just natural variation in our field. So because we weren't starting at that same starting point between drained and undrained plots, we also looked at how PMC was changing over time. So comparing each of the subsequent 6 sampling events to the fall 2019 sampling event. And with that, we still did not see any significant differences, and it didn't change how we interpret the data. So water extractable organic carbon. So this is a proxy for active carbon. It's an easily mineralizable food source that is loosely bound. It's organic molecules loosely bound to soil particles, and those molecules are soluble in water. And so again, WEOC is not the only active carbon, but what we expect to happen in drain plots, we expect WEOC to decrease for similar reasons to PMC. As we create likely better conditions for microbes, they are going to be decomposing that carbon. How is it measured? Pretty straightforward. Soils are shaken and centrifuged, and then that leftover or that water is filtered just to make sure we have those suspended particles removed, and then the water is analyzed. So what do we see? We see again with this similar trend to PMC that WEOC is decreasing with depth and over time. What do we see is significant? We see a sampling event is significant with that trend decreasing over time. Unlike PMC here, we don't see any of those differences between drained and undrained plots when we look at drained and undrained compared at a given depth in a given year. So what is kind of the main takeaway from these two microbial active pools? It's that they weren't influenced by drainage like we thought they were going to be. So kind of shifting here and then now looking at POM and Mayon, our short and long-term carbon storage. So POM would be on this chart, we would see POM as kind of this active carbon and Mayon as the stabilized or old carbon. But shifting over to this image, we can see POM on the left and Mayon on the right. And so how are these two fractions different? POM, as you can see, is that least decomposed plant material that can be physically protected in aggregates. Mayon on the right we see is of microbial origin. So it's those microbial byproducts or leached compounds that are protected through mineral associations. And then subsequently, because of those of that composition, we see a carbon and nitrogen ratio being similar to either those starting plant materials or to microbes. So with PMC, or not PMC, with POM, we see larger C to N ratios on the scale of 10 to 40 and more variable. Mayon we see on the range of 8 to 13 and less variable. And so kind of looking at their location and abundance, in soils, the majority of soil would be classified as Mayon. And so with POM, we see higher concentrations in surface soils and decreasing with depth. Mayon, we see that other trend where it's increasing in concentration with depth. So what do, or how are these measured? Again, it's a size fractionation. So we see POM is greater than 53 microns, and on that right image that is in the bottom row of pans, Mayon is less than 53 microns, and that is on the top row of pans in that right image. And so soils are shaken and sieved and then oven dried, and those pans are really the result of what we're looking at. Well, this is kind of getting into how we're characterizing these, but the carbon content, so carbon was determined from combustion analysis. Again, pretty standard. Soils are finely ground to what is pretty much a powder and then combusted. So how are we characterizing changes in POM and Mayon? So all of these are going to be in terms of POM, but you could just switch out Mayon and it's the same thing. So we're looking at quantity, and this is really getting at that location and abundance in soils. And so this would be thought of as gram POM per gram soil, or POM as a percent of soil mass. Quality, this is looking at the organic carbon to nitrogen ratio and also the amount of the organic carbon content in each of these fractions. So gram organic carbon per gram POM or percent organic carbon in POM. And then finally, the relative contribution. So how much soil organic carbon is made up of organic carbon from these two fractions. And that would be gram POM organic carbon per gram soil organic carbon. Okay. So first, looking at POM and Mayon quantity, or that, well, this is really getting at location and abundance in soils. We see greater POM in drained plots from 0 to 30 in 60 to 90. And so all of these graphs, we're going to look at POM on the left and Mayon on the right. And I will reiterate this every slide, but sometimes the ranges on the Y axis will change. But we see those 3 years in which they're sampled across all 3 depths. And we see a greater POM again in 0 to 30 centimeters and 60 to 90 in those drained plots. And we see this general trend that POM is relatively less of the soil mass than Mayon. In Mayon on the right, we only see a significantly greater Mayon in undrained plots from that 60 to 90 centimeter depth. When we're looking at quality or that organic carbon in these two fractions, we're looking at the same range on the y-axis. So comparatively in the same range for both POM and Mayon. With POM, we don't see any significance at any depth for any variable. But we see this general trend that we have more organic carbon in the POM located in surface soils compared to those of greater depths. In Mayon, you can probably see the trend and significance. We see this significant decreasing trend of organic carbon content in Mayon from 30 to 60 and 60 to 90. When we look at the organic carbon to nitrogen ratio, again, we're looking at different ranges on the y-axis. POM, we're looking a lot, we're in that range of 10 to 40 that we expect. Mayon in that range of 4-ish to 15. So we see POM being having an organic carbon to nitrogen ratio more indicative of crop residues, and Mayon more similar to microbes. And so again, in POM, we don't see any differences at any of these depths, but we do see this general trend of increasing with depth. In Mayon, we see a significant interaction from 0 to 30 centimeters between treatment and year, and then from 30 to 90 centimeters, again, those two deeper depths. From 30 to 60, we see this slight uptick in the organic carbon to nitrogen ratio, followed by a decrease. And then in 60 to 90, we see this no change from 2019 and 2020 down to a decrease in 2021. If you're interested in statistics, those letters for 30 to 60 would be AB, AB. And for 60 to 90, 2019 and 2020 would both be A's, with 2021 being a B. So 2019 and 2020 being similar, and then a decrease in 2021. And finally, the relative contribution of these two pools to soil organic carbon. Again, we are looking at different scales on the y-axis, with POM being overall less to total soil organic carbon. If you look, these numbers don't add up to 1. This is due to the analytical variability of measurements, and because we have such low organic carbon content across the board in our samples. And I'll touch more on that a little bit when we get to organic carbon stocks. But here we see in POM from 30 to 90 centimeters, we see a significant treatment and an interaction of treatment in year. And then in Mayon, we see year is significant at all depths, and a significant interaction from 30 to 60 centimeters. So that's kind of a lot of information, but what are really kind of the key takeaways from this that I'm going to focus on in the conclusions? Well, it's that POM organic carbon contributes relatively little to soil organic carbon. And also notably that we saw greater POM quantity in drained plots at 2 depths compared to undrained, which we were really not expecting. So soil organic carbon, and so this is kind of looking at these organic carbon stocks. Again, we expect that they are going to decrease in drain plots due to increased mineralization. So how are organic carbon stocks calculated? that organic carbon content is multiplied by bulk density for that given depth. So bulk density, we see bulk density was greater in 2021. And you'll notice here that we only have bulk density for 2 years. We don't have bulk density for 2019. If you remember, that was a wet fall, and there is a time crunch to even get the tile drain installed. So bulk density did not happen. When we look at organic carbon stocks, those 2019 values were used, or they were calculated from 2020 bulk density. So when we look at organic carbon stocks, we again see this trend with greater values in surface soils and decreasing with depth. And so again, these stocks are on the low side of what we would expect, and that's from that low organic carbon content. And so nothing from the field history that we have looked through explains this. And I believe we have field history until from the 1970s. Yeah, the 1970s. So the past 50 years, there's nothing in that that's really indicating why this would be. This is just what we have. And so organic carbon stocks is typically how people look at organic carbon in soils. And so what do we see here? From 0 to 30, a significant interaction. And then when we look at 30 to 60, we have a significant year, treatment, and interaction between the two. When we look at organic carbon stocks between drained and undrained plots and how they differ in a given year at a given depth, we again see one difference, and this is in 2019, which again, we expect to be natural variation in the field and not a treatment difference. So what's kind of this takeaway here? We're not seeing a strong influence of drainage on these organic carbon stocks. And finally, everybody's favorite greenhouse gases. We're looking at these as a way to look at microbial byproducts. We're only looking at carbon dioxide. So as a recap, carbon dioxide is produced from the aerobic decomposition of organic matter. And again, if we shift microbial communities and those conditions for them, we're going to expect shifts in their byproducts. And so we expect that with increased decomposition in drain plots, we're going to see an increase of carbon dioxide emissions in those drain plots. So how are these sampled? We use a flux chamber approach. We use 5 chambers in each of those 4 plots that you can see in that top image. The soil bases are kept in the soil, or the chamber bases are kept in the soil across the growing season. And then those caps are, they create a tight seal as you attach them. What do we see? We see across 2020 fairly similar trends. And then in 2021, our drought year, we see fewer emissions. And so when we look at this, we see significantly fewer emissions in 2021, but not between drained and undrained plots. When we look at cumulative emissions, we see, again, greater emissions in 2020 compared to 2021, with undrained plots trending slightly higher in 2020 and undrained trending slightly higher in 2020 and drained trending slightly higher in 2021, but comparatively similar. And so this main takeaway from this last part with greenhouse gases is that we're not seeing an increase of carbon dioxide emissions in drained plots. Okay, conclusions, home stretch. Okay, what does all of this mean? So when we're looking at PMC and water extractable organic carbon, again, they weren't influenced by drainage, but rather time. And so what does that really translate to? That result, microbial processing and easily mineralizable food sources changed with seasonal and annual shifts, not drainage. And so why might we expect this? Well, when you think of these two being pools influenced by microbes and those changes occurring in the soil across the growing season with crop production, we're going to see shifts. When we see discrepancies in trends, or maybe we're not following the same pattern between PMC and WEOC, we know it's just reiterating that WEOC is not the only food source that is accounted for in PMC, or the only microbial food source. And then finally here, since these two pools, these labile carbon pools, have been shown to indicate how bulk carbon is changing in soils, since we're not seeing changes across these 2 years, maybe in the short term, we wouldn't expect to see changes based off of these results. So PMC and WEOC as soil health indicators. So these two metrics are part of the Haney soil health test. And this next part is really estimating nutrients available in your soil for microbial assumption, or for microbial consumption. And so throughout the growing season, carbon and nitrogen are going to mineralize, and it's important. I've given no love to nitrogen, but it's important to know that they don't cycle independently. And so they can actually, PMC and WEOC, along with WEON, or water extractable organic nitrogen, can be used for agronomic decisions such as fertilizer recommendations. And so this is based out of Ward Laboratories in Nebraska. But this calculation is, oh, there we go. This calculation is based on WEON as an easily mineralizable nitrogen source, but also how active your soil microbes are. So not only do you have to have nitrogen in your field, you have to have something to mineralize it. And so ultimately that water extractable nitrogen in parts per million is from a known range is scaled up into what is likely to mineralize in your soils. And so we see it in spring 2020 and spring 2021, this wide range of what we would expect based on our data. And so this kind of just reiterates that we know these pools shift over short periods of time. But if we're going to use indicators in this way, there has to be, again, not really a great standardization in these measurements sometimes. So you have to really consider the depth to which you're sampling, the time which you're sampling, and then this is really getting at that spatial variability that we can have in fields. So shifting to carbon dioxide emissions, so we didn't see significantly more in drain plots. Why is that? Well, for two reasons. We don't see significant differences in labile carbon, those PMC and WEOC values between drained and undrained plots. So in WEOC, we're not seeing a difference in the food source. In PMC, we're not seeing a difference in how those microbes are processing the carbon. And then again, with soil moisture, going back to this, we're seeing fairly consistent conditions with some gaps here and there. So again, back to Palm and Mayon, what do all this mean? What does all this mean? And without splitting hairs between every little change in trend in graphs, we see between composition, CETA, and ratio, location, and abundance, we see mostly what we expect for the most part, except that we see Palm contributes relatively little to overall soil organic carbon. So why is this important, or what is the takeaway? Well, because Mayon relies on mineral surfaces for those organic particles to adhere to, we see that at some point Mayon is going to saturate. So in this image, you can see Mayon on the left, Palm on the right. And so as we build organic carbon in these soils, at some point, we're going to run out of mineral surfaces for those particles to adhere to, and Mayon is going to saturate or plateau in these soils. So what does this really mean? Well, increasing soil organic carbon may only be possible with POM at a certain point. And so how can we see that as being possible based on what we saw and that potential to increase POM? We saw POM quantity was greater in drained plots, again, at those two depths, 0 to 30 and 60 to 90, while POM quality, that organic carbon content, was not changed by drainage. So the potential for increasing organic carbon accumulation is really through increasing just the quantity of POM. And finally, organic carbon stocks. When we look at this, we don't see a difference in how soil organic carbon stocks are changing between drained and undrained plots. So this might indicate that in the short term, we're not seeing these differences. This might not be an abrupt, quick process, but really, what effect did the drought have when the main thing we're looking at here is drainage, and half of the entire second year, half of this study, we didn't use drainage because we didn't have enough water. So what's next? Looking at carbon in the long run, and Anna and Lindsay, you're gonna do it. So stay tuned for years to come. But yeah, we will see how these soils respond after a drought, and what if we start to see significant changes with time. And then you might also be thinking, well, what about nitrogen cycling in drained plots? And so you should join Aaron Prinkel's defense tomorrow at Tuesday, June 7th. Aaron has not seen the slide before. I told him it would be debuted here. So this is an S415 or over Zoom, different Zoom link. I will be admitting people in Aaron's room. So if I get emailed that you joined my link, I will send you a quick email. But he is looking at nitrogen cycling, soil physical properties, and biological indicators. Funding, thank you to Minnesota Africa Agricultural Fertilizer Research and Education Council and Minnesota Corn Growers Association for funding this. Thank you to Anna and Lindsay, my two advisors. Certainly grad school has not been the easiest over the past 2 years, doing my entire degree during COVID, but they have been so great and supportive. Thank you to John and Jess, my committee members, for your support and guidance. And then John and Jess, in addition to Josh, Galen, and Carol, for all of the problems or data issues that have been figured out. And then the Pease lab group, people who have handled samples or kept me sane over the past 2 years. And that is it. So I guess questions, let me pull. Okay, so I can see people. Or feel free to unmute, or do people have that option? I'm guessing they do. Or you can put it in the chat. Or I can just stand up here and drink coffee. I'm sure my committee will appreciate that. Yeah. Any questions about coffee? John.
So what are you doing next?
Oh, yeah. So I this summer will be moving out to Davis, California, and I'll be working with ARS as a biological science technician. Start date TBD. Yes, Brent. Oh,
Hey, Kyle. Nice job. I enjoyed that talk a lot. I'm glad you put up that figure from the Co2 paper, and that got me to thinking a little bit more about like where we would expect to find pools of organic matter that would be vulnerable to these changes. And right in high clay soils like this, you've got a lot of available surface area, and so the organic matter that's left after decades of management is probably pretty tightly associated with those mineral surfaces and really well protected. And so how do you think that these results would be different if you were there to start this experiment at year zero, when the landscape was first transitioned to cultivation, or maybe if we were in different soils that had lower clay content?
okay. So you're asking how going back to cultivation, how would we expect that to look compared to now?
Yeah, I think maybe what I think my point is that these soils in this location are probably some of the most robust in terms of the organic matter that's left, just by virtue of the high surface area and the long management history. And so I wonder if there are other places that you think might be more sensitive to the kinds of measurements that you would make.
More places as in different different systems, or?
Sure, different soils, different starting, pre-settlement or pre-cultivation land cover classes.
Yeah, I guess something I think about a lot is, well, it's something that maybe one slide touched on, but disturbance. And so I always think of disturbance as when we just rip up the soil and install these tile lines. But I guess looking at systems that are typically undisturbed, tend to be, I guess from what I have read, I think of, decreased disturbance, more stable. So I think of, up by Duluth where we have all these forests. Yeah.
Yeah, I mean, I think you're right. I also think that, less disturbed soils are probably places where there's, high organic matter stocks that aren't protected, or they're not protected as intimately, with the soil minerals. They're protected more just by virtue of, good aggregate structure or palm, as you've shown here. So, yeah, no, nice job. I enjoyed the talk.
Thanks. Okay, so question here from Jeff Fetch. Did you take soil samples between the tile lines? Yes. So we had a little difficulty early on with GPS and exact locations, but yeah, we took samples between the tile lines, never on top. Yeah. Yeah. I don't think so. We're also contaminant deeper soil depths. So yes and no. I'm thinking of, and I'm sure everyone in my lab group knows, there's one point that just has extraordinarily high POM. That is about 40% of the soil mass. That is, I can scroll up to what that looks like. That is this point here. That is just always high. So potentially, potentially. Yeah, Josh.
Hi, Kyle. Great presentation. Thanks. I was curious about the bulk density measurement. So it looked like in your later years, there was quite a jump, especially at depth in the bulk density. Yeah, is that, I'm curious about that. Is that, what are your thoughts on that increase in bulk density, and how do you think that might have influenced results in any way, if at all?
Yeah. So we had this discussion in my practice whether or not these values passed the eye test, and they did. So they are within reasonable range for what we would expect. Mm-hmm. How this then impacts results. So, and this is something Anna and I have thought about, but when we ultimately convert this into organic carbon stocks, we see differences in bulk density, obviously from 2020 to 2021, but then we also don't have bulk density for 2019. And I'm thinking of one specific comment that was, we'll see if we get pushback on this, but I guess I don't, in that first year, how much do we expect bulk density to change? I guess we thought not a lot, but then we see this jump from year 1 to 2.
Yeah, that would be my expectation too, is that you might not see a whole lot in the short term, but I've never done a drainage study like this either. And yeah, so. Have you looked at the raw concentrations at all, just to, I mean, and done some analysis there to help, you know, sort of tease apart that influence of the bulk density if you don't have it for 2019, right?
Yeah. So I don't think I have it as an extra slide in here. Mm-hmm. No, but they are fairly similar to this trend we see with organic carbon stocks. But yeah, I can go back and look at that, especially probably at that top depth where it's more variable. Because I remember I see it in my head, and it's that same trend at the lower depths where there's not a lot of variation, but I'll go back and look at that.
Yeah, just something to consider, but great job overall, Kyle. Great presentation.
Kyle, this is Satish Gupta. I got a question for you. Could you go back to the slide on bulk density, please?
Yes.
You did a good job in the presentation. It's really well done. I was just curious. If you look at 2021, your bulk density is getting some values of over 2.5.
Yes.
You know why at even 2? That's pretty high for those soils.
Yeah. So this is something I've looked into but haven't found anything concrete. So I don't know that there's a question, but I'm going to ask you a question.
All right.
What effect does the drought of 2021 have on bulk density?
Well, you know what the particle density of the soils is?
The particle density. No.
Okay, it's 2.65. That
Okay.
means you disperse a particle in the water. There's no air in there.
Okay.
So by each individual particle. So if you pack it very tightly, get everything out, then you'll get close to 2.65. And I'm surprised that your advisor didn't look at 2.65 is the particle density of more soils. Unless you got very high organic matter content, then it'll go down. So you're already close to the particle density. That's not possible. Okay, so how did you make the bulk density measurements?
So they were done with the hydraulic Giddings probe,
Yeah.
measured and then oven dried.
How did you get the volume?
The volume we know based on the size of the core and the length. Is that the volume?
And the cores were how long?
So they were these segments of 30 centimeters. And the diameter of the core fairly small, 3 centimeters.
We use the wider core for these.
Oh, right. We weren't up there.
Yeah.
Oh, the 3-inch cores. Oh, okay.
I mean, the bigger the better for doing.
Yeah. Okay. I don't know if you heard, but 3-inch cores.
And they went down to 60 to 90 centimeters.
Yes.
I think these values are pretty high. So there must be even 2 is very high. There must be something going on that for those soils. So you guys may want to look into it. Overall, you did a good job. Very nice. Did you also measure any carbon coming out of the drain water?
We did not.
Okay, maybe I think the variability in these soils is so much that it'll be very hard to look for small changes on organic carbon or particulate carbon in 1 or 2 years. It's going to take some time. And it might be a good idea to see how much of the carbon is going out. I think Brent had done some work a long time ago. He can probably help you get you some answer. Anyway, good job. Thank you.
Thanks. That's okay, Josh. Heidi, over time, do you think that subsurface drainage will help slow carbon's release to the atmosphere in Minnesota? I think that's a loaded question. Again, based off of this study, that's 2 years, where we're just starting to formulate trends and where we have 2 very different years. I guess it's hard to say, but I mean, if we keep seeing droughts, maybe we won't release so much greenhouse gases. Not really great, or yeah, I guess that's the trade-off.
We wrap up, if people can follow up over email if they have some questions. Thanks again.